Everyone, happy new year. I hope 2023 is off to a great start and no one is feeling too nervous about biology. I know I've disappeared yet again for quite a long time, but please, this is most likely going to be the norm now as I have entered into a very busy phase of my life. It's been well, I didn't expect that it would be this busy, but it has been, but it has also been very fulfilling. So I am not complaining at all, but I do want to say I apologize sincerely to those who bought the textbook, the workbook that I put out on Gumroad and Amazon last year and expected the solutions to come immediately after. That was my plan as well. I thought, oh, the solutions will come right after, but really that week was a week where I just got pulled into very busy projects, and since then I haven't been able to get some time off. But this year is starting off a bit lighter, so I'm hoping to get the solutions out. I've told myself before your June exams, but I'm pretty sure it'll be out before then, like way before then, because I'm really committed to getting this done. I want to say thank you to all of you who have supported by purchasing the workbook. I hope you're finding it useful, and all the little nuggets of information I hope are very helpful for you. For those who were able to bag the workbook for free, I guess you're glad, and I'm glad that you took that on. I made some copies available for free just for students at the beginning of the year as a way of saying Happy New Year, and I made it a pay what you can. So I mean, some students have said, okay, I'll pay £2 or £5, and that's been much appreciated. Thank you. They're limited copies, so if you haven't gotten one, if you can rush over, you might be lucky. I can't say, but once those copies are done, then it's going back to the normal price that I put it out for. So I will get into the chapter 1 solutions for the workbook today, and I'll try to make this as quick as possible. I'm hoping to do this in less than 12 minutes. All right, let's do it. Okay, so this is the first question in the workbook where you basically get to practice your conversions. And the numbers I've given in the workbook are the ones in black, and I simply tell you to convert these to the units that are at the top. So the easiest one obviously is that when you're going from millimeters to micrometers, you're multiplying by 1000. And when you're going from micrometers to nanometers, you're multiplying by another 1000. There is another unit that comes after nanometers, and it's picometers, but you hardly get questions about like that go all the way to pico. But if you were to get that, it's also just by another 1000, right? So don't fret too much when you're seeing that. You know that when you're going from meters to meters, that's times 1000. And when you're going from meters to centimeters, it is times 100. Okay, so these already should give you an idea of how to approach this. And basically if you're going to work backwards, like I gave you this number here, 570 at the bottom, you're working backwards, so it means you just keep dividing all the way until you get to meters. And obviously you need to be careful when you're going from centimeters to meters, because that's where 100 comes in and it's not 1000. Something else I want to point out on this is that if you look at the way I've written my numbers, I've written them out in full, but you don't have to do this. I wrote this out just to show those of you who might have an issue or a struggle with this, how you can count the number of zeros to determine the exponential point. So in this case now we have 6 zeros here, so you can simply just count 1, 2, 3, 4, 5, 6. And if we're putting our decimal there, then we know that we can write that as 22 to the power of 6. Okay, but if we decide to move it one place up, it's going to be 2.2 to the power of 7. When the zeros come before the decimal, as they all do in this case, then the exponential is a positive one. I hope this makes sense. But if it comes in this case where the zeros are after the decimal, I suppose that's the best way to put it, so you're counting this way, then you're going to write this, for example, it will be 1, 2, 3, 4, okay, so we can say that's 57 to the power of minus 4. So it becomes negative because of the way we count. So, okay, a good way to remember it rather is if you're counting backwards, so if you're counting from right to left, then it's a positive exponential, and if you're counting from left to right, it's a negative exponential. So just bear that in mind. Okay, and this one is a question to help you practice magnification. I think I put quite a few there. Again, I've written out the explanations for how you should approach it. So if you just want to pause that and read through, that's absolutely fine. But what I just want to stress on this slide is that whenever you're given a scale bar like this, and you have a number on top of it, that number is the actual size of the object, okay? And in order to do the magnification calculation, you would have to measure the length of the scale bar, okay? So you measure the length of the scale bar, and whatever the length gives you in millimeters, because you're using a small ruler, you convert that. Okay, that just looks like a wiggly worm for some reason. I meant to write double M, so like that. My pen can be a bit funky sometimes. So in millimeters, that's what you get when you measure this with a ruler. So you simply multiply it by 1000 to get it to micrometers. The reason why you have to get it to micrometers is because the value on top here is in micrometers. If you divide the millimeter value by the micrometer value, your answer will certainly be wrong. So you want to ensure that the unit you take here for the length of the scale bar is the same unit as what you have on top. Now, in a case, and I'm just going to use this as an example, in a case where you are not given 50 micrometers, right? This number is not here. But they tell you, for example, that the magnification of the object is 100 times. So if you use M divided, M is equal to I over A, you know that in this case you're saying 100 is equal to I divided by A. A is the one you don't know because it's not given, okay? So I, again, is the length of the scale bar converted to micrometers. For this answer here, you might not get the exact same answer as I did because I measured this from my screen. So for some of you, I mean, depending on the Zoom levels or whatever it is you're working with, the answer might be slightly different. But I want you to understand the principle that whenever you're given a scale bar, you measure the length of the scale bar, you convert it to micrometers, and then you divide it by the value that is here. The length of the scale bar is your image size, and the value on top of it is your actual size. All right, this is another very easy one. It's a bit of a direct math problem. So it's like if the eyepiece graticle in a light microscope is 100 units in length, and that's usually what it is, about 100 units, and it covers 0.5 millimeters on the stage scale. So that basically means from 1 to 100, I'm saying 1 to 100 units is equivalent to 0.5 millimeters. Okay. Then the question is, what is the size of a cell that measures 25 units on the eyepiece graticle? So if 100 units is 0.5 millimeters, then you obviously know that 1 unit is 0.5 divided by 100, and 25 units would be the result of that times 25. So that would be your answer in millimeters. And then you have to convert it to micrometers because the question I asked you said to give your answer in micrometers. This is a similar problem to the one we looked at just a little bit a while ago, or a short while ago rather, where you're basically again calculating the magnification from the scale bar. Always remember that the length of the scale bar is your image size, the number that is on top or underneath the scale bar is your actual size, and you can calculate magnification based on that. In this case, if your answer is a decimal, check the question. If the question says give your answer to the nearest whole number, do not present the decimal, just round it up and give the whole number. If the answer doesn't say anything like that, then you can present the decimal. And always remember that when you write magnification, there's an X that is behind it, or you can even put the X in front of it. So you can write that as 605. I was about to write 650. So this would also be marked correctly. But without the X, your answer might not be marked correctly. Okay, this is one I really wanted to touch on because I know students tend to make mistakes here. So whenever you are given a group of cells like this and it's a photomicrograph and you're told to calculate the magnification and you've been given the actual length, then whatever it is that you measure here will be the image length or the image size. When measuring this, always make sure that you are using the points that have been marked within the question. So in some cases, they might say marked, like they might mark this point and say from P to Q, and they draw a line from those two. That's what you should measure. So the length is not from here to here, okay? That's not the length. That's what you might refer to as the breadth if you're into math and geometry and shapes and so on. That's what you might refer to as the breadth. The length is this, from this point to this point. So more or less like the longer side, if you want to think of it that way. That would be the length. So make sure that you're measuring that and you pick a specific cell that they would have mentioned for you. Don't just go pick a random cell, because as you can see, all the cells have different sizes. So they have based their answer on a specific cell length, and so they would expect that you get the same answer. Okay, this is a very good one. So it says basically the stage scale of a microscope has divisions of 0.01 millimeters. What is the size in micrometers of a cell that covers 40 units on the eyepiece graticle? So for those students who are not aware, when you look into the microscope, you see a scale, okay? There's a scale inside the microscope that is part of the microscope. And this is an activity I used to do with students whenever they started biology in my class. I'd tell them to look into the microscope and there'd be a slide there with bacteria or plant cells, and they would be excited and they just never noticed the eyepiece scale. So this is the eyepiece scale, and then you get another scale that looks like this, and they'd tell you, your teacher or your lab technician would tell you what the divisions are on the scale. So in this case, it's told you that 40 units, but it's asking you what is the size of a cell that covers 40 units. You don't have to do a lot of math here, because if you look at how they are aligned, already it's telling you that the divisions on the stage scale, that's the 0.01 millimeter divisions, are actually covering 40 units. Okay, because if you look here, this is 10, all right? That's 20, that's 30, and that's 40. So you know that any cell that covers 40 units here has a size of 0.01 millimeters. And if you want to give the answer in micrometers, you simply multiply that by 1000. Please always read the instructions, especially when it has to do with the units, to see what the question is asking you to present your units in. Okay, assuming the question in the cell in the question above has a magnification of 100 times, what is its actual size? So because you're looking at the cell through the microscope, this answer you have here is the image size, okay? Because you're seeing an image of the cell, not the actual cell. So in that case, you know that if magnification is 100 and your image size is 10, your actual size would then be 10 divided by 100, and that will give you 0.1 micrometers. Okay. Then there's a question here about the difference between magnification and resolution. I'm not going to go over that because that's pretty straightforward. But here, in terms of labeling the cells, I just wanted to point out a few things that you might find useful. So over here, whenever you see something here that looks like how you would draw the sun as a child in art class, that's the centrism. Okay, so that's something to just bear in mind. That is the centrism. Whenever you have an oblong cell like this and it has a zigzag inside of it, that is your mitochondria, okay? This is the Golgi body, and what you can see with the Golgi body is that it has all these units that are not really joined together, okay? So that's the Golgi body. These very dark dots, and they're usually presented this way, are the ribosomes, okay? This is the cytoplasm. This is the lysosome. So if you see a round vesicle that's like standing by itself, you can consider that to be the lysosome. And in some cases, even if you say vesicles, they would still mark it correctly. But most of the time when the vesicles are presented in the cell, they are shown next to the Golgi body. So this would, for example, be a vesicle. Okay. My writing is just so terrible with like computed stuff. It's hilarious to see. And then yeah, just what else do I need to tell you? Okay, yeah. Also know that the nucleus and nucleolus, right, are separate from each other. So don't get confused when you see arrows pointing to these two. And when you see an arrow pointing right to the bottom here, to the last thing, that's not the nucleus. That would be the nuclear membrane. So there's a difference between this pointing there and pointing in here and that pointing in there. So please make sure you have those in mind. Oh, and then it asks what kind of cell is shown in the image above or in this image. That's definitely an animal cell. And how do you know that? The shape of the cell, all right? Plant cells have a very distinct rectangularish shape, while animal cells tend to be a bit oval, sometimes round, sometimes oblongish, but they're not necessarily as fixed as a plant cell. You can also point to the absence of a large vacuole, the presence of a centrosome, the absence of chloroplast. All of these are features that enable you distinguish an animal cell from a plant cell. And this is what the plant cell is. So I think the key things I'll just point out here is when you're looking on the outside of the plant cell, please take note of where the arrows are pointing, especially with regards to the plant cell. The externalmost part, like the most external part, not the externalmost, that's not a word. The most external part is the cell wall. The cell membrane is the one that's on the inside, okay? So the cell membrane is the one on the inside, while the one on the outside is the cell wall. Bear in mind that the membrane surrounding the vacuole is called a tonoplast. And also remember that the vacuole with plant cells rather is rather large, so that's the vacuole over there. And you can see the vesicles now next to the Golgi body. And with the chloroplast, it usually looks like you have a stack of gold coins or a stack of coins arranged inside of them. That's how you know what the chloroplast is. Which cells or which organelle would you typically find in a plant cell, in an animal cell but not in a plant cell? Usually centrioles. And what is a virus? So here I have underlined, and I hope you can see it clearly because now that I'm presenting it, I can see that I didn't do an exact great job, but this is the definition for a virus. It's an acellular organism. Please always remember to mention that it is acellular. And it has genetic material as well as a protein code. And I've underlined those two because those are usually the two keywords they look for when they're marking this question. So this is just a list of all the different functions of the cell organelles. I'm not going to go through them. You can pause on this just to see what I have said and check if it correlates with what you have in your workbook. But I see no reason why this would be a miss for you because you can simply go into your textbook. The reason why I make students go through this is because I want them to always remember what each organelle does, because as you know in CIE you don't get questions that necessarily ask you for functions of things like this, but it will be woven into a question that requires you to think deeply about the function of that organelle. So just go through this by pausing it. So it continues here, and I think the big thing that you might not be aware of might be the cilia and the microvilli. The microvilli are used to increase surface area, so you would find them, for example, in your gut, where they increase the surface area to absorb nutrients from the food that you eat, and also to house the bacteria that help you process your food. And the cilia are like tails or extensions from the surface of the cell, and the cell sort of uses them to swim forward. This again, I'm not really going to go through because again, you can just pause it. This is just for you to know the differences between prokaryotes and eukaryotes, because at this level of biology, those are the only two types of cells that you're expected to know, that they are prokaryotic cells, which are typically things like your bacteria, and they're eukaryotic cells, which are things like your fungi, your plant cells, and your animal cells. And I think this is the last question where I gave you some information in the workbook on fractionation, and then I said, in what order would the organelles in the cell emerge if you were running a fractionation experiment? And this is the order. What you should bear in mind is that the ribosomes are the smallest, and the nucleus is the biggest, or is the heaviest in the cell. Everything else is now dependent on their weight, right? Chloroplast is heavier than mitochondria. So if the question is asking you about the fractionation of a plant tissue, then you want to mention that the chloroplast will come out after the mitochondria. But for the most part, this is simply the order that it will come out in. So I think that's it. For those of you who don't have the workbook, you can always get a copy on Gumroad. I think there's still a few free copies that you can grab. So if you're lucky, you might be able to grab it. But if you're not, then you will have to pay for it. Not might, you will have to. Yeah, thank you all so much. I thought this would be a quick thing, but I see that I've now gone for 20 minutes. And I guess what I can say is biology is fun. So time flies when you're having fun, right? So for that reason, biology is fun. That is a mathematical derivation I have just done. Have a good time, everyone. Until next time. Bye-bye.